Oops. Hope it's working okay. Can you hear me there?
Yep, you're fine. You're
okay.
Good.
Welcome, everybody. Thanks for being here on Friday. Today we have a very special speaker, Dr. Jing Chen. She's a colleague of mine in the Department of Biological Sciences. Jing is a mathematical biologist. She has a really highly interdisciplinary educational path. And you will see that in her presentation as well. She got her bachelor degree in biology in 2002 from Fudan University in China, which is one of the top ranked universities in China. After that, she decided to travel a little bit around the world and did her master degree in mathematics in biosciences in 2004 in Germany at the Technical University of Munich in Germany. In 2010, she earned her PhD in biophysics from UC Berkeley. And after that, she moved to a 5 years postdoctoral position at the National Heart Lung Blood Institute, where she was a very prolific researcher. Jing's interest revolve around what is an interesting question to answer using mathematical modeling. So she builds mathematical models for what she considers intriguing biological problems, especially in problems that involve temporal, spatial, and/or mechanical dynamics. Her research has spanned through a number of really cool topics in biology, including mitotic signaling, spindle assembly, bacterial mobility, microbial population dynamics, and today, circadian rhythms. So you will see one of her very latest adventures today. So she really tries to contribute to biology by really providing us with some models that help us to pose and test provocative questions. So without further delay, I'm gonna let the screen to Jing. Thanks a lot for joining us, and I'm really happy that you accept our invitation, Jing.
Thank you. Thank you very much, Carla, for the really nice introduction. And good afternoon, everyone. It's my pleasure to give this talk about one of the latest adventures in my group, like Carla just said. And in this talk, I'm going to show you how mathematical modeling can provide insights into an interesting aspect of the circadian rhythm. So speaking of circadian rhythm, probably everybody here has heard of the term at some point in your life. So essentially, our body has this internal biological clock that helps us anticipate and adapt to day and night cycle. And this can be manifested as fluctuations in our hormone level. Like you can see on this cartoon, cortisol release peaks at around early morning, and melatonin secretion peaks around late night. And our body temperature and blood pressure also fluctuate over the daily cycle, and eventually our behavior also fluctuates. So for example, we have the highest alertness during the morning, and then in the afternoon, it turns over to best coordination and fastest reaction time. And then in very early mornings, we have the deep sleep. So basically, the circadian rhythm tunes our body and our physiology such that we can perform the right way at the right time over the daily cycle. So maintaining a normal circadian rhythm is important for our health. If the clock is disrupted, we'll get sick. So jet lag is probably the most common symptom when our internal clock does not match the cycle of the environment. But of course, jet lag is harmless and transient. But if a person's circadian rhythm is constantly disrupted, it would make damage to the health. So for example, shift workers have higher risk for metabolic diseases, which is caused by constant disruption of their circadian clock. So some audience may have noticed this little footnote for the figure. Yes, I downloaded the figure from the Nobel Prize website. And the study of the circadian rhythm by these three great scientists won them the 2017 Nobel Prize in Physiology or Medicine. And in fact, the middle guy, Michael Rosbash, gave a VTLSS seminar here in the year 2017, just a few months before he got the Nobel Prize. So I feel really excited and honored to be giving a talk in the same seminar series on a related topic. So the work for the Nobel Prize is about how the rhythm is generated. So what they found is basically this molecular clock mechanism that exists in every single cell. Yes, every single cell in our body has this clock ticking. And this molecular clock consists of a number of core clock genes that form a regulatory network. And this cartoon is only showing the most central ones in the clock mechanism. So these core clock genes, they form feedback loops. So for example, these BMAL1 and clock, they're transcription factors that can bind with regulatory elements called EBOX, and the EBOX can control the expression, the transcription of other core clock genes, including PER, CRY, reverb, and RAW. And PER and CRY are particularly important here. They accumulate in the cytoplasm during the night, and during the day, they enter the nucleus where they can bind BMAL and clock and inhibit their activity. So this way it forms a negative feedback. So PER and CRY essentially inhibits their own transcription. So RAW and reverb controls the transcription, and BMAL, they also form feedback loops. So among these feedback loops, the negative feedbacks are especially important, and they are essential for generating the oscillation. So this principle that negative feedbacks generates oscillation actually has been long known by modelers. Here's just a list of a small fraction of modeling papers about how negative feedbacks generates a circadian oscillation. And the earliest paper published was even before any of the core clock genes has been identified. So over the decades, as the experiments tell us, more and more people have constructed increasingly realistic model on this topic. And our group has also recently submitted a paper about this core circadian clock, which is listed here at the bottom. But I'm not going to talk about it today. Instead, I'm going to talk about what happens downstream of this clock. So far we have a fairly good understanding of how the core clock proteins become rhythmic. But how does the rhythmicity translate to rhythmic functions of cells, tissues, organs, and the whole organism? Turns out this is rather poorly understood, and this is what I'm going to talk about today. So this cartoon actually shows the tip of the iceberg. So you can see a lot of these core clock proteins are transcription factors. So for example, BMAL and clock, they're transcription factors that binds with EBOX, RAW and reverb are transcription factors that binds with an element called RRE, and there are also a couple of other transcription factors in the core clock mechanism that binds with other types of regulatory elements. And so these core clock transcription factors, they control the expression of CCGs on this cartoon, and the CCGs stand for clock control genes. And CCGs are actually way more than the 4 genes shown on the cartoon. There are hundreds of genes in the cell that contain these elements that can be transcriptionally regulated by the clock transcription factors. Okay. So rhythmic transcription seems to be an obvious answer for rhythmic gene expression, right? So if we think about the central dogma, the gene is transcribed into mRNA, and mRNA translated into protein. And so if we have a clock transcription factor that makes the transcription rhythmic, then mRNA would become rhythmic, and then protein would become rhythmic. Bingo, we have solved the problem, right? Now, well, I wish the problem was that simple. So it turns out the rhythmic control of gene expression is much more complicated. In fact, gene expression itself is much more complicated than the central dogma. So this cartoon is, so both transcription and translation consist of many, many sub-processes. And this cartoon is actually illustrating these sub-processes in a simplified version. So rhythmic transcription factor, which we discussed on the previous slide, is just one part of this one tag. And there are evidence showing that all these other sub-processes can also be rhythmic, including chromatin conformation, RNA splicing, mRNA nuclear export, mRNA accumulation and degradation, translation, and protein accumulation. Okay, so all these other sub-processes can also be rhythmic. And today, my talk will focus on a process that is implied but not explicitly labeled on this cartoon. I'm going to talk about these little AAA poly A tails of the mRNAs. As you can already see on this cartoon, their lengths can vary. And in fact, their lengths are dynamically regulated and they are shown to be rhythmic as well. So why do we care about the rhythmic regulation of the tail of an mRNA? So to see why, let me briefly introduce what the tail does in gene expression. Basically, the poly A tail is a ubiquitous and versatile regulator of the mRNA. So it's ubiquitous because except for histone mRNA, all mRNA in the eukaryotic cells have a poly A tail. So these mRNAs first obtain their poly A tail during transcription. And once the mRNA enters the cytoplasm, the length of the poly A tail can be dynamically regulated. And the poly A tail is a versatile regulator of mRNA because it organizes molecular machineries that can regulate mRNA at nearly all stages of its life. Particularly, the poly A tail is involved in controlling mRNA degradation and translation. So this is why poly A tail and its rhythmicity is important for rhythmic gene expression. So the evidence for oscillation in poly A tail length was discovered by my collaborator Shihoka Kojima, who is a colleague of mine in the same department. So she did an experiment where she took samples of the mouse liver around the clock, and then she extracted the mRNAs from the mouse livers, and she designed a method to separate the mRNAs from the same genes into two groups, a group with long tails, long poly A tails, and a group with short poly A tails. And then she can use the ratio between the abundance of the long-tailed mRNAs and the abundance of the short-tailed mRNAs as a quantifier for the poly A tail length. So from these experiments, she identified over 200 mRNAs that exhibit rhythmicity in the long and short ratio in the poly A tail length. So the long and short ratio oscillate over the daily cycle. And then she compared these mRNAs to those that are rhythmic in the total abundance, the L plus S level. So the two categories only overlap partially. And very interestingly, she found that the protein rhythms are closely correlated with the poly A rhythms, especially for mRNAs in this group, okay? So which are the mRNAs that are rhythmic in the poly A tail length, but steady in their mRNA abundance. So even if their mRNA abundance is steady, many of them exhibit protein rhythms. And this suggests that in these cases, the protein rhythms likely derive from the rhythmicity in the poly A tail length because poly A tail regulates translation. Okay. So this being said, we want to understand how the poly A tail is dynamically regulated and gain rhythmicity and how that affects rhythmic gene expression. So we approach this problem using a mathematical modeling method. So this work was done by my graduate student, Xiangyu Yao, and we started with a simplistic model like this. Okay, the model is built upon what we know about the life cycle of the mRNA poly A tail. And we follow the example of Shihoko's experiment and designate two subpopulations of mRNAs, one with long tail and one with short tail. Okay, so first the transcription produces long-tailed mRNAs. And this comes from the observation that nascent mRNAs typically have long tails. In mammalian cells, nascent mRNAs typically have a poly A tail of 200 nucleotides. And then in the cytoplasm, the long and short mRNAs can interconvert between each other due to the deadenylation and polyadenation processes. And then the model also assumes that degradation only happens to the short-tailed mRNAs. This is because in the canonical mRNA degradation pathway, which is the pathway that most mRNA uses for its degradation, this pathway requires the poly A tail to be first shortened to a critical length, which is typically just a few nucleotides. So that's why we assume that only the short ones can be degraded. Then we can write two differential equations to describe the dynamics of this model system. So the first equation, dl dt, describes the rate of change of the L population. So L increases due to transcription, decreases due to deadenylation, and increases due to polydenylation. And the ds dt equation keeps track of the rate of change of the S population. So it contains two equal and opposite terms for deadenylation and polydenylation, and then an extra term, negative term for the degradation. So to depict the rhythmic control of all these four processes, transcription, deadenylation, polydenylation, degradation, for each of these kappa, the rate terms, we assume that they follow a cosine function of time, which is defined by the mean rate, K, amplitude A, and the peak phase phi.
Sorry, Jing, can I ask you a quick question?
Yes.
And I apologize for my ignorance, but is there one deadenylate enzyme, or there are multiple deadenylate enzymes?
Enzymes, you're asking about enzymes, right?
The enzyme that shortens the tail is only one deadenylation enzyme, or is there multiple deadenylation enzymes?
This is very mouthful. Yeah, there are actually multiple deadenylases, and there will be a later slide talking about this.
Okay.
Okay, so in the model, we're starting to investigate the dynamic behavior without really looking at which enzyme is doing the work. We're just looking at general quantitative principles.
Okay.
Okay. All right. So we have the equations, and so using the equations, we can put in the mean rate, peak phases, and amplitudes of these four processes, put it into the model, and the model will predict the corresponding mean levels, peak phases, and amplitudes of the L and S population. And we're particularly interested in these three output quantities, namely the LS ratio, which, like Shihoko's experiment, quantifies the poly A tail length, the total mRNA, which is just L plus S. And we're also interested in the long-tailed mRNA abundance, which is L. We consider this L as a very rough quantifier for mRNA translatability, because poly A tail organizes the translation machineries and so regulates the translation process. But I'm saying the L is a very, very rough quantifier by any means, because the relationship between the length of the poly A tail and translation efficiency is indefinite, and it can vary a lot from gene to gene. So I'm saying this is very rough, but because the model is very simple, so this is our best choice in terms of associating some model quantity to mRNA functionality. Okay. So yeah, the model can, predict, what kind of input gives what kind of output. But the problem is we don't know the parameters for most of the genes. And the parameters are likely going to vary a lot from gene to gene. So there is a very strong uncertainty in the input parameter values. So in this case, what can we learn from the model? What does the model tell us? So then we realize, okay, the actual valid question we can ask using this model is which rhythmic regulator is most important for the rhythmicity in poly A and mRNA? And this question can be answered by parameter sensitivity analysis. So through parameter sensitivity analysis, we can understand how each of the output quantity on the right depends on each of the input quantity on the left. And taken together, this will tell us, which process on the left is most important for which quantity on the right. Does that make sense? Of course, I'm not going to get a response in the online. So if we can answer this question, we'll be able to obtain a general understanding of the rhythmic dynamics of poly and mRNA, even if we don't know the parameters for specific genes. Okay, we can understand the general principle here. All right, so now let's think about the parameter sensitivity analysis. Actually, the problem we just talked about remains a challenge for the parameter sensitivity analysis, which is that the input parameters can span over a broad range for different genes. The peak phases can be anywhere between 0 and 24 hours. The relative amplitudes can be anywhere between 0 and 1. And the mean rate spans at least over 2 orders of magnitude. So what are we going to do with such a large variation in the input parameters? So because these parameters are very uncertain, local parameter sensitivity analysis, which is the typical way how people do parameter sensitivity analysis. So these analyses are done around a fixed parameter set. And so their results would become meaningless because whatever we conclude from one parameter set may not be true for another parameter set. So if we want to understand the general principle, the general behavior of the model, we need to examine the parameter sensitivity for all possible parameter values. And this is called global parameter sensitivity analysis, and which is typically done by running the model with randomly sampled parameter sets throughout the whole range of possible parameter values. So we tried that. We ran the models with randomly sampled values for all these parameters. And then we can compare each pair of input and output and see how they're related in a general sense. And a straightforward way to visualize the results is to draw the scatter plots. So here I'm showing the scatter plots for one of the output, which is the phase of the LS ratio, the quantity that we use to indicate poly length. So we have scatter plots of this output with respect to the peak phases of all the 4 input processes: transcription, degradation, deadenylation, and polymorphism. So from these scatter plots, we can already see some patterns. We see that apparently the phase of the LS ratio most strongly depends on the phase of deadenylation because the data are concentrated within 2 narrow stripes. So here I want to point out that these 2 stripes should really be considered as one continuous stripe because the phase is a circular variable. So 0 hour is equivalent to 24 hours. So, you should really consider this plot as, as something that can loop around. So this stripe below can be, cut and paste over the stripe above to form a continuous single stripe. So in other words, the relationship between the phase of the LS ratio and the phase of deadenylation is really monomodal, not bimodal. Okay, we actually only have 1 stripe. It looks like 2 stripes because phases are circular. So it kind of breaks over. So besides the phase of deadenylation, the phase of polymorphism also seems to control the phase of LS ratio, but to a lesser extent. But the other 2 phases seem not to have much control over the phase of LS ratio. So scatter plots are great, but we would like to be able to quantify these results as numbers for easier comparison, okay? So the first thing we tried is to calculate the correlation coefficient between the inputs and outputs. And we tried 2 commonly used correlation coefficients, the Pearson correlation and the Spearman correlation, and they actually have very similar results. So for the same data sets that I showed on the previous slide, the same 4 scatter plots, the same input phase of LS ratio and the 4 input phases. So we can see some problematic results. So for the correlation between the phase of LS ratio and the phase of deadenylation, okay, so this is the one with the strongest pattern, the correlation coefficients are negative, even though the scatter plot seems to shows a positive slope in the pattern. Okay, so apparently the correlation coefficient algorithm must have calculated the relationship, you know, in a way that's perpendicular to the stripes instead of parallel. So that makes it a negative correlation instead of positive. Okay, so that value makes you scratch the head. But what probably you can argue that, okay, maybe we can just look at the absolute values. The absolute values are still the largest for this group of data. However, if we look at the next group of data, even the absolute value would fail. So this is another result where I'm comparing the same 4 inputs, but the output is now the phase of the L plus S, okay, and a different output. So you can see from the scatter plot that the phase of the L plus S is most correlated with the phase of transcription. However, the estimated correlation coefficient has the lowest absolute value. So it means that absolute value is also no good for quantifying the relationship between the input and output. So basically the correlation coefficients, this method, can generate very misleading results if we're analyzing circular variables. So this happens because the method of correlation coefficients has an intrinsic underlying assumption for the linearity between the X and Y data. But circular variables are intrinsically nonlinear, right? As I said, they loop over, so they're intrinsically nonlinear. So when you're using a method that has an assumption about linearity on highly nonlinear data, you will generate misleading results. So we need a better method. And then we learned about a method called the Sobel indices, which is a variance-based sensitivity analysis, which does not assume linearity between the data. So what is variance-based sensitivity analysis? So remember that we're running the models with randomly sampled parameters, and there are a variance both in inputs and outputs. So the variance-based sensitivity analysis tries to decompose the variance in the output Y as many terms, which are variance contributed by single parameters, X1 or X2 or X3, so on and so forth, plus the variance contributed by interactions between 2 terms, X1, X2, X1, X3, etc., etc. And then the variance contributed by interactions among 3 parameters, 4 parameters, so on and so forth, until we reach the variance contributed by interactions among all the parameters. Okay? And the Sobel index is defined as the proportions of these decomposed parts. So the single Sobel index is the proportion of the variance contributed by a parameter xi alone over the variance in total variance in output Y. And another Sobel index called the total Sobel index is defined as the proportion of the sum of all the variance terms with a contribution from xi over the total variance in output Y. Okay, so by these definitions, we can see for each pair of input parameter X and output quantity Y, there's one single Sobel index and one total Sobel index. The total Sobel index is always larger than the single Sobel index. And the larger these Sobel indexes are, the more the input parameter influences the output, right? So their variance takes up more weight in the variance of the output. And so the output Y is more sensitive to this particular parameter X. Okay, so the value of the Sobel indices gives us a quantitative index of how X and Y are related. And because the variance-based sensitivity analysis does not make any assumptions about the linearity of the data, So it's very useful in analyzing parameter sensitivity for nonlinear models. And when we implement this method on our model, the results look beautiful. Okay, so I'm again showing you the same data sets that I used before. So, and I'm comparing the results from the Sobel indices with the results with Pearson and Spearman correlation coefficient. So you can see that now the Sobel indices are the highest for the pair between for the phase of LS ratio and the phase of deadenylation. So it correctly, consistently represents the strong dependence between this pair of input and output. And for this other set of results where the output phase is the L plus S, now the Sobel indices for the phase of transcription are the highest, which is consistent with the pattern we see in the scatter plot. Okay, so we have a very good method using which we can quantify the parameter sensitivity in a global sense. So let's come back to this workflow which I showed before. So we want to identify which rhythmic regulator is most important for the rhythmicity in poly and mRNA. And the method we're going to use for the parameter sensitivity analysis is the Sobel indices. And let's look at the results. So for the phase of LS ratio, which is the output quantity that indicates the poly A tail length, we can see that the Sobel indices for the phase of deadenylation are the highest. Okay, and then this is followed by the phase of polyanylation. So basically this says that the phase of the LS ratio is most sensitive to the deadenylation phase and a little bit less to the polyadenylation phase, but nearly nothing to the transcription and degradation phase. And if we look at the Sobel index result for the amplitude of LS ratio, we can see a consistent conclusion. So the Sobel indices for the amplitude of deadenylation are the highest in this case, followed by the amplitude of the polyadenylation. But the transcription and degradation processes contribute negligibly to the amplitude. So both results consistently show that deadenylation is the strongest contributor to the rhythmicity in the LS ratio or poly tail length. Now let's look at how the total mRNA abundance depends on the parameters. So if we look at the phase of the total mRNA abundance, L plus S, we see that the Sobel indices for the transcription and degradation, Sobel indices for the phases of transcription and degradation are the highest. But the phases of deadenylation and polyadenylation also contribute a little bit. Okay, so all four processes have some contribution to the rhythmicity in mRNA abundance. And for the sake of time, I'm not going to show the Sobel indices for the amplitude anymore. And last, we're interested in the quantity L, which is the very rough quantifier for mRNA translatability. So for L, the Sobel indices are the highest, again, for the phase of deadenylation, followed by the phase of polyadenylation. So this is the same pattern as we saw for the LS ratio. So far, our model has revealed a general pattern, which is that the deadenylation is the strongest contributor to the rhythmicity in poly A tail length and the rhythmicity in this rough quantifier for mRNA translatability. So next, we're going to bring this model a step closer to real data. We're going to combine this general quantitative principle that we learned with some real data. So in Shihoko's paper about polyrhythmicity, she reported rhythmic expression of the genes that encode the deadenylases and poly A polymerases in mouse livers. And these are the enzymes that carry out the deadenylation and polyadenylation reactions that Carla was asking about. So what's interesting is that 5 deadenylases are shown to be rhythmically expressed, and their peak phases are concentrated in 3 narrow windows around hour 2, hour 5, and hour 13. Meanwhile, only 1 poly-A polymerase is rhythmically expressed, and it peaks around hour 3.5. And so we're wondering, so why do the deadenylases want to peak in 3 windows while the poly-A polymerase only has 1 peak? So to explore this question, we did an in silico experiment. So where we changed the distribution of the peak phases in each of the 4 processes and examined how they affect the distribution of phases in the 3 output of interest. So for the first case, we assigned the peak phases in all 4 processes to be evenly distributed around the clock. And for this case, we get evenly distributed phases in the outputs. This is kind of no-brainer, and it's just our control experiment, if you wish. Then in the next 4 cases, we make, so in each of the next 4 phases, we make one of the processes peaking in 3 narrow time windows, while keeping the other processes having the evenly distributed phases. And so when we plug in these phase distributions into the model, this is what we get. So you can see that only for case 4, where we have the deadenylation peaking in 3 distinct windows, we can get LS ratio and the quantity L peaking in 3 distinct windows. So in other words, having 3 peak windows for deadenylation has a very strong clustering effect on the phases of L over S ratio and the phases of L. And this clustering effect is not seen in any of the other cases where the other processes peaks in 3 distinct windows. Okay? So now if we plug in experimentally observed distributions, what are we going to see? So here, the deadenylation and polyadenylation, we just assume their phases follow the same pattern as the expression of the enzymes that I introduced on the previous slide. And for transcription, it turns out transcription phase is also not evenly distributed around the clock. So there is a science paper showing that nascent transcription mostly concentrated between hour 12 and hour 18. So we took that data and plug in this phase distribution for the transcription. There's not any data about the phase distribution of the degradation. So we just assume, leave it as an even distribution. Okay, so with these experimentally observed phase distribution, we can still see 3 distinct time windows for the phase of the LS ratio and the phase of L. Okay, so the clustering effect remains with the experimentally observed data. So here I want to emphasize that except for the phases, the amplitudes and the mean rates are all random in all cases. So this very strong clustering effect indicates that the rhythmic deadenylase can cluster LS ratio and L, the rhythms of LS ratio and L, regardless of rhythmicity in other processes. So it's a very robust clustering effect. And this clustering effect is essentially a synchronization effect, because you can imagine that the genes that are controlled by the same deadenylase would have synchronized rhythms in poly-A length and mRNA translatability, right? They would fall in the same narrow window. So this is a very interesting finding, and we wonder, could it play any potential biological role? And to enable this discussion, I'd like to zoom out and bring your attention to the organization of circadian clocks in our body. So as I mentioned before, every single cell has a clock, but the cellular clock is very noisy. So the cells in our body needs to communicate with each other to stay synchronized. So there's a specific region in our brain that serves as the central pacemaker. Okay, this region is connected to our eyes and aligns its own rhythms with the light and dark cycle. And then downstream, it sends signals throughout the body to synchronize the peripheral clocks, which are the clocks in these other tissues and organs. Okay, so, but very interestingly, even though, all these peripheral clocks are entrained by the central pacemaker, it turns out that the circadian gene expression still has very strong tissue specificity. So what do I mean by tissue specificity? So among different tissues, there are actually very little overlapping between rhythmic genes. So genes are rhythmic in one tissue, may not be rhythmic in another tissue, and there are actually very small overlapping between rhythmic genes. And also the same genes can have different rhythmicity in different tissues. They can peak at different times in different tissues. At the same time, the expression pattern of deadenylases are also tissue specific and can differ quite significantly across different tissues. So combined with our finding that deadenylase could have a very strong synchronization effect on circadian gene expression, we wonder if these tissue-specific deadenylase activity may play a role in mediating these tissue-specific rhythms in gene expression. Okay, so that's one speculation that we have, which is motivated by our model finding. So another speculation is related to this food shown on the cartoon. So actually our bodily clock does not only entrain to the light and dark cycle, it also responds to other systemic signals, and feeding is a well-known example. So feeding is known to influence our circadian rhythm, and that's why on transcontinental flights, meals are served at local times, because they can help you adjust to the new time zone. So it turns out that rhythmicity of some deadenylases are controlled by the feeding rhythm, not the light cycle, not the central pacemaker. Okay, so they directly respond to these feeding signals. So then again, combined with our finding of the synchronization effect by the deadenylases, we wonder whether these deadenylases may play a role in entraining the circadian clock to the feeding cycle. So make a mechanistic connection between, feeding and, rhythm adjustment. Of course, all these are speculations, and I'm curious how future studies in the field will tell us about these speculations. And finally, our SOBO index analysis can also suggest underlying factors for observed rhythmic patterns. And here I'm just going to talk about one example for two classes of patterns observed in Shihoko's experiment, which she named as the class 1 and class 2 mRNAs. So in the class 1 mRNAs have a rhythmicity in transcription, total mRNA abundance, and poly A tail length. But the class 2 mRNAs have steady total mRNA abundance, but they still have rhythmicity in transcription and poly A tail. So so our question is to ask, what are the parameters that makes the difference between class 1 and class 2? So we want to find out the major contributors to the difference. So because the difference between the two classes is whether the total mRNA abundance is rhythmic. So we try to look at the SOBO indices for the amplitude of the total mRNA abundance. And apparently, the results tell us that the amplitude of the L plus S is most strongly dependent on the mean degradation rate. So if we compare the histogram of parameters that gives the class 1 behavior, the purple, and the parameters that gives the class 2 behavior, the green, we can see that the mean degradation rate tends to be significantly higher in class 1. And this is actually consistent with the experimental observation. So Shihoku's paper reported that the mRNA half-life is much longer in the class 2 mRNA. Then we can see after this mean degradation rate, there are 3 other parameters that also have some moderate contributions, which are the amplitudes of the transcription and degradation and the mean rate of deadenylation. So if we look at the histograms for class 1 and class 2 parameter sets in these 3 parameters, we can also see a significant difference, which is consistent with what the subway indices tell us. And another interesting observation of the subway indices result is these 2 phases, the transcription phase and degradation phase. They both have very high total indices, but vanishingly small single indices. Okay, so remember the total indices indicate the contribution from a parameter when it's interacting with other parameters. So the fact that these 2 parameters have very small single index and very high total index indicates that they only affect the output, the amplitude of L plus S, when they're interacting with other parameters. But they're the only 2 parameters that show this pattern, show this contrast between single and total. So we speculate, okay, perhaps they're interacting with each other to affect the amplitude of L plus S. So if we look at a histogram for the phase of transcription minus the phase of degradation, bingo, we can see a very significant difference between the 2 classes, which indicates, okay, it's really the difference between these 2 phases that, that contributes to the amplitude. Okay, so this is a little bit of a technical description of, what the subway indices results can tell us, how, what kind of information we can interpret from this kind of result, this kind of net analysis. All right, so with that, I would like to conclude the talk. So hopefully I have convinced you that deadenylation, according to our model, is the dominant contributor to the rhythmicity of poly tail length, and possibly that of mRNA translatability. So deadenylation is our champion here. And because of the strong effect by the deadenylase, they could possibly synchronize this rhythmicity of the poly tail and mRNA translatability, regardless of other rhythmic inputs. And this may serve some important functional roles. And lastly, the variance-based global parameter sensitivity analysis is a powerful tool for analyzing highly nonlinear models. And so next time, if you're running into a similar question with highly nonlinear data, you can try variance-based global parameter sensitivity analysis. So I would like to thank the students the people who did the work. These are all the people in my group, and Xiang Yao is the graduate student who did the work. And I would also like to thank my collaborators, Dr. Shihoku Kojima, produced all the data, and Dr. Shi Chen is another faculty member at Virginia Tech who actually taught me about the subway indices. And Ben Heiderecht, the undergraduate students in the lab, and Dr. John Tyson and me together worked on the model for the core circadian clock. So if you're interested, you can also check out that paper. And our group is financially supported by a seed grant from Fraley Life Science Institute and recently by NIH. So I would like to thank you for your attention and I'll welcome questions.
Thank you very much, Jing, for presenting. That's a really cool story. I followed this for a really long time. We have a couple of questions. Before we jump on that, I'm gonna take advantage of me being the first one here. Do we have any experiment actually where there is a knockdown of of any deadenylating enzyme, and you actually show that when this enzyme is knocked down, you actually change the phase of the long versus short ratio?
I'm not aware of that, and I mean that would be a super cool experiment, and I really look forward if somebody would will do that experiment. Maybe it's maybe the latest papers might have reported something in that line. I haven't yet followed.
No, it's okay. I guess it's probably a difficult experiment to do as well. We have a couple of questions. Let's start with Charlene. He says, in your model, you consider the only long tail poly A is directly synthesized from transcription. Can short tail poly A be directly synthesized from transcription as well?
Usually not. So the polyadenylation mechanism inside the nucleus is separate from the polyadenylation mechanism in the cytoplasm. So the polyadenylation inside the nucleus happens co-transcriptionally. So there's a very specific machinery, doing that, and it typically produces a long tail, and this is generally true for, throughout the genome.
We have another question. This time is from our colleague Lina Ni. Is it possible that a messenger RNA poly A tail is shortened? Translation is decreased, but mRNA is not degraded.
It's shortened.
Yeah.
mRNA is not degraded, but translation is down, right?
Yeah.
Yeah, it's possible. There are actually more complicated mechanism in the cytoplasm that regulates the mRNA activity. So for example, there are what people call, these granules called P-bodies and stress granules, where the mRNAs can be transiently stored, where the translation is suppressed, but it's a reversible process. That when the condition changes, the granules can disassemble, and then mRNA can be reactivated. So yeah, so that's possible.
We have one more question. This time is from Daniela Cimini. She says, I noticed that in your in silico experiments, you could reproduce the 3 peaks. Mm-hmm. However, the peaks occur within time windows that did not match the experimental observations. Could this be due to degradation peaks within certain time windows instead of occurring at constant rates throughout the day? If so, could you adjust that in the model to make the 3 peaks match the experimental observations?
Oh, so Danielle is asking, why do I have 3 peaks not at the same? Mm-hmm. For case 2 through 4, we're just trying to do a general in silico experiment to see what happens if you have 3 distinct windows, and we're just taking a generic pattern to prove the principle. And so in the final case, we plug in the experimental data. The question is a little bit long, and I think I might have missed some of the remaining parts.
She meant in the last column.
In the last column, yeah?
Yeah, if you look at the very last column, mm-hmm. She says, the peaks occur within time windows that did not match the experimental observations. Could this be due to degradation peaking within certain time windows instead of occurring at constant rates throughout the day? Your very last column.
Degradation peaking within peaks occurred within time windows do not match the experimental observation.
I see. Danielle is asking about why these 3 peaks do not look at the same time as these 3 peaks. So actually deadenylation inhibits LS ratio and inhibits L, right? Deadenylation brings L to S. And so it's expected to have the opposite phase. So technically speaking, this window around 12 should correspond to this window.
okay? And this window around 2 probably correspond to this window. And this window around 5 correspond to this one.
Does that make sense?
Daniela?
They're supposed to be anti-phase because deadenylation antagonize LS ratio and L.
Okay, and we have one more question. It's also from Charlene. Cells have many deadenylases. Only several of them have circadian rhythms. How do they coordinate which other to regulate deadenylation and make poly-A tail rhythmic?
Oh, that's a great question.
That's a great question.
So you can see that I have, in my in silico experiments, I have actually made a simplified assumption. I assume that the deadenylation activity and the polydenylation activity follows the same pattern as the expression of deadenylase and poly-A polymerase, which may not be true, right? So you can imagine, a gene could be simultaneously deadenylated by 2 deadenylase from different groups, right? That peaks at different times. Then what, right? But of course, that's we don't know about that, and so our model is just trying to do like a proof of principle exploration, and so I think the Xiaolin's question will have to be answered by future experimental study.
Yeah, I think so too. Okay, if there's no any further questions, let's thank Jing for her presentation, and congratulations on your NIH grant.
Thank you.
And I'll see you next Friday. Okay, see you guys.
Thank you.
Yeah. Thanks, Jing. Thanks a lot.
Thank you very much, Carla.
No problem. Great talk. Thanks.